Hi, so those of you who don't know me, I think there's several of you out there. I'm at Colorado State University, so my lab's been open for about 2 years now, and we use really a variety of methods to look at how different three-dimensional chromatin structures function, really in repressing transcription. We do most of this work in the context of quiescent budding yeast, and so today I'm going to tell you how I use mostly genomics in order to establish this kind of twist on the regular yeast model as a good model system for looking at different levels of chromatin structure. My voice is kind of going a little bit, so sorry, everyone. It's good. All right. And so I'm going to skip over my normal introduction of 3D chromatin structure and just go right into why yeast. And so a lot of what we know right about two-dimensional chromatin structure comes from budding yeast. Now, many factors and processes are well conserved between yeast and human cells. Additionally, yeast have a small genome, which makes doing very high resolution genomics financially feasible. So we can do a lot of different samples and we can sequence very, very deeply. And of course, the awesome power of yeast genetics means it's very easy for us to either delete or mutate factors of interest to see what happens, basically. So it's a very easily perturbable system. Of course, however, there's a problem looking at budding yeast in the way they're typically looked at in lab settings. And that is that cycling yeast have a pretty open and very transcriptionally active chromatin structure, which is not ideal for looking at how 3D chromatin structure represses transcription, right? And so to get around this, I've adopted a budding yeast model, and this yeast is in a state of quiescence. And so quiescence is a reversible state in which the cell really exits the cell cycle in G1 and suspends the majority of cellular processes. So these cells are not really transcribing or really doing much at all. And yeast can actually stay in this quiescent state for thousands of years. But human cells also do this. It's important for health. Now, what's distinct about quiescence from other G0 states is that it is reversible. So even though these cells can exist in this dormant state for extended periods of time, they can also re-enter the cell cycle and resume normal proliferation. In the lab, how this works is that cells are grown in culture for at least a week. During this time, as they exhaust glucose in the media, a subset of these cells enter into quiescence. And so these cells build up their carbohydrate stores and adopt a very stress-resistant and long-lived program, whereas the cells that fail to enter quiescence basically experience widespread genome instability and die. And so what's really nice about this is that these quiescent cells actually become more dense than the non-quiescent cells, which means that we can purify them out on density gradients and get large amounts of pure quiescent cells for experiments. And so what is really interesting about this system for my purposes is that if you look at the total number of actively transcribed genes between cycling cells, or log as I'll call them, and quiescent cells, you can see that transcription really goes off dramatically in these quiescent cells. And then the genes that are on actually tend to be different than the ones that are on in cycling cells. Now, at the same time, if you do DNA DAPI staining and measure the total volume of chromatin in these cells, you can see between G1 arrested cells and quiescence, the total volume of chromatin decreases almost by half. So this is really telling us that there may be something very interesting going on at the 3D chromatin structure level, mediating this interesting thing we see going on with transcription. And so really my goal as a postdoc, and really this continues to be the main goal in my lab, is to look at how different three-dimensional chromatin structures change between log and quiescent cells to mediate this change that we see in cells being mostly transcriptionally active to cells becoming mostly transcriptionally repressed. And so today I'm going to talk about chromatin structure at two different levels that's involved in this process and that changes in these cells. So we're going to look at chromatin loop domains and then chromatin fiber structure. And at the end, if I have time, I'm going to talk about some of our current projects in the lab. So the main way that we look at chromatin structure in 3D in cells is through micro-C. So I'm not going to go through all of this for this group, but essentially this is a very high-resolution version of high-C. So it differs from high-C in a couple of major ways. The first is it employs these long cross-linkers in addition to regular formaldehyde cross-linking, and so it's better at capturing longer range interactions. Additionally, instead of using restriction enzymes to digest chromatin, it uses micrococcal nuclease, and so it digests down to single-nucleosomes, which means we can get single-nucleosome resolution from this. And because we're doing this in yeast, with small genome size, we can actually sequence deeply enough to get not just single nucleosome resolution, but really excellent coverage of each single nucleosome, so we can be very confident in looking at these single nucleosome interactions. Here's an example looking at sort of the difference between micro-C and high-C, in case anyone hasn't seen it. So here we're looking at a region of one chromosome, and essentially we're zooming in more and more and getting to higher and higher resolution. And essentially at all resolutions, micro-C really does a better job of capturing chromatin interactions than high-C. All right, so when I did micro-C initially in log and quiescent cells, one of the sort of most obvious things that came out is the presence of these dots in the quiescent cell data located pretty far from our local diagonal interactions. And so as everyone here probably knows, this kind of dots tend to indicate the presence of these long-range chromatin loops, typically stabilized right by some kind of chromatin structure or some kind of protein. And so these loops look suspiciously like tags, right, that have been well characterized in metazoans. And so the tags, of course, are mediated between interactions between cohesin and this transcription factor CTCF. However, these kind of loop domains really had not previously been identified in budding yeast, who additionally do not have CTCF. However, yeast do have three loop extruding SMC complexes. So that's cohesin, condensin, and SMC56. So to see which of these might be involved in forming these loop structures, I basically did ChIP-seq and log and quiescent cells looking at these. For the purpose of time, I'll just tell you I couldn't find any evidence that either cohesin or SMC56 were really bound to chromatin at all in quiescent cells. However, when I looked at condensin, I saw something really interesting. So here I'm showing the total number of ChIP-seq peaks between log and quiescent. You can see that during this kind of transition, the number of condensin peaks goes up about threefold. If we go on and look more specifically at different locations where we see condensin, here I'm showing metaplots. We're looking at different regions, and what we see is that during quiescence, condensin moves from its normal locations at tRNA genes, centromeres, and the rDNA to bind upstream of coding gene promoters in quiescent cells. And so we see particular enrichment at divergently oriented coding genes. I lined up this condensin ChIP-seq data with the micro-C data. So here these lines are indicating the boundaries of these chromatin loop domains. And you can see that these condensin ChIP-seq peaks line up pretty perfectly with the boundaries of these loops. This is again supporting the idea that it's condensin that are making these loops in these cells. And so we next wanted to really confirm this and see what happens when condensin is depleted. So we came up with a way to conditionally turn off the expression of condensin and repeated these micro-C experiments. So here I'm showing condensin depleted micro-C data subtracted from the wild type quiescent micro-C data. So contacts that are up in the wild type are in red, and contacts that are up when we get rid of condensin are in blue. And essentially what we see is that when we lose condensin, all of these loops and all of the structure within these loops is red. So we lose this when we get rid of condensin, indicating that we're losing these loop structures and moving to a more extended chromatin structure. When we look at transcription by RNA polymerase II ChIP-seq, we can see that about 20% of all genes in the genome turn on when we get rid of condensin, and the genes that had previously been on actually come on even more. And so frequently what we see is that we have a really strong condensin binding in between the boundaries of two of these loop domains. And in many cases, these are located right at divergently oriented genes, in which one gene is on, so it has RNA polymerase, and the other gene is off. When we get rid of condensin, we lose interactions within these domains. We gain interactions across the boundaries of these domains. And in many cases, we gain Pol2 at the gene that had previously been off on the other side of this boundary. And so kind of the model then is that between log and quiescence, condensin relocates throughout the genome where it forms these large chromatin loop domain structures. And it seems to mostly be responsible for repressing the transcription of genes located right around the base of these loops. And so we really next begin to wonder, so if condensin is responsible for repressing transcription of these genes, sort of what is repressing the transcription of genes located within these loops, which really is the majority of genes in the genome. So we thought maybe something going on with the local chromatin structure might be responsible for this. So that's the second kind of level I'm going to talk about, is looking at chromatin fiber structure in these cells. And so to do this, we turned back to the microC data and looked a bit more closely at what was going on at interactions right along the diagonal. So here is microC data looking at a small region of chromosome in log. And here we're looking at the same region in quiescent cells. So you can really clearly see that the interactions along the diagonal increase pretty significantly in quiescent cells as compared to log. So that's consistent with compaction really of local chromatin structure. And so one of the nice things about this being microC data is that we can actually go in and specifically look at the different patterns of nucleosome interactions that we see. And so you can imagine if you have a very extended chromatin array, most of your interactions will be between a nucleosome and the nucleosome right next to it on the DNA strand. If you have something that's more compacted, you'll have interactions between a nucleosome and nucleosomes that are further away on the strand. And so we did this analysis of the data. That's essentially what we saw. And so for looking at our log data here in purple, you see most of our interactions are between a nucleosome and the one right next to it. And these interactions decay pretty rapidly. When we look at our quiescent cell data, we see that there really is an increase in sort of longer range nucleosome interactions at this local scale. So again, that's consistent with sort of local chromatin fiber compaction in these quiescent cells as compared to log. We also collaborated with Tamara and Stephanie and her lab to do mesoscale modeling using our genomics data. So this is using MNA-seq data to get nucleosome positions, and then looking at linker histone binding and histone acetylation from ChIP-seq. So here we're looking at a 40 kb region of chromosome 1. Here's sort of a representative structure in the log case. And we look at the same thing in quiescent cells. You can see the modeling really predicts that these quiescent cells are compacted locally, which again is very consistent with our microC data. So one of the really cool things about this modeling is that Stephanie was able to go in and look at all the different kinds of interactions that were present in these structures. One thing that Stephanie found is that there's an increase in interactions between histone H3 and H4 tails and the cores of other nucleosomes. And so this was very interesting to us because, as I think probably everyone is well aware, it's been shown for a long time looking at reconstituted chromatin in vitro that extended nucleosomal arrays compact into sort of 30 nanometer fiber structures in the presence of divalent cations. And it's long been thought that this interaction really has been mediated between interactions in the H4 basic patch on the H4 tail and the acidic patch of a nearby nucleosome. And this has been shown by many different groups. Additionally, as was shown really nicely in Craig Peterson's lab before I got there, was that the acetylation of a single lysine residue in the H4 tail, lysine 16, is really sufficient to disrupt this compaction in vitro. And one thing that we knew about quiescent cells before I started working on this project is that the histone deacetylase complex RPD3 is really targeted across the genome in these cells where it massively deacetylates H3 and H4 tails. And so one thing that is happening between log and quiescence is that histones are going from being mostly acetylated to completely deacetylated. And so putting all of this together, we wanted to look to see if perhaps the H4 tail basic patch was involved in regulating the local chromatin compaction that we see in these quiescent cells. And so to look at this, I generated a series of yeast strains with different substitutions in these residues in the basic patch. And the first thing that we did is this very low resolution method where we do DNA DAPI staining of cells and look at Z stacks by confocal microscopy. So you can measure the total volume of chromatin in cells. We do this for hundreds of cells so we can get good statistics. So when we look at wild-type quiescent cells, this is the chromatin volume that we see. When we add in a single alanine substitution of lysine 16, we get a significant increase in chromatin volume. When we do the glutamine substitution, so kind of the acetylation mimic, looks very similar. When we substitute the two arginines here, we get an even larger increase in chromatin volume. And when we do all 5 residues of the basic patch, we get this really large increase in chromatin volume. So this is consistent with, again, the H4 basic patch regulating chromatin condensation and quiescence. And this is something that happens only in quiescence. This does not happen in log cells. And so we went back to microSEE data to look at these mutants at high resolution. So here we're looking at wild-type quiescent data. And here we're looking at data in quiescent cells in this double alanine substitution. So you can see we do lose a lot of our interactions here along the diagonal in these mutants. And this is looking at the 5 residue mutation. You see it's even more so that we lose contact. When we go in and specifically look at the pattern of nucleosome contacts that we see essentially when we mutate these residues in the H4 tail, we lose a lot of these kind of longer range but still local nucleosome interactions. Again, consistent with this mechanism being important in these cells. And so I think this is really nice because this is telling us that even though kind of compacted structures that we see in cells probably aren't these perfect 30 nanometer type fibers, the mechanism regulating these is absolutely the same mechanism that was predicted for 30 nanometer fibers from all those in vitro experiments. So these experiments are very valuable. All right, so we then looked at transcription in these different H4 mutants. So here we're looking at Pol2 ChIP-seq data, and we're looking, in case you're not familiar with these heat maps, each of these lines represents a single gene, and these are oriented with the transcription start sites in the middle, and we're looking plus or minus 1 kb on either side. And so we're looking at all genes in the yeast genome, and this is our scale, so purple indicates there's transcription. In wild-type cells, we don't have a lot of transcription, right, because they're quiescent cells. We then add in increasing numbers of substitutions to the H4 basic patch. We see that transcription really increases proportionally relative to the number of substitutions that we introduce. And so really in this most extreme kind of mutant here, about 40% of all genes in the genome turn on. And again, this is something that's specific to quiescent cells. Log cells don't care about this. You can get rid of the H4 tail, basically, they don't care. And so what this is telling us is that in between log and quiescence, deacetylation of the H4 tail drives an increase in interactions on the same DNA strand, which leads to this local compaction of chromatin fibers. And so, as many people probably know, one of the big questions in sort of the loop extrusion field is really understanding how extruding factors are able to kind of make these loops in the context of chromatin when there are all kinds of obstacles present on chromatin in the cell. And so we thought this was a nice opportunity because we have a condition where we have an extruding factor that's doing something important, and we also have this local change in chromatin structure that we can perturb by mutation. And so we wanted to see if changes in the local chromatin structure actually affect the ability of condensin to extrude loops in cells. And so here I'm showing wild-type quiescent cell data, and we've lined up the condensin ChIP-seq data underneath. And so we do the same thing looking at one of our H4 tail mutants. We can see the formation of these stripes in the mutant microSEE data, and these stripes line up pretty perfectly with the location of condensin ChIP-seq. And so these stripes are generally thought to indicate the presence of active loop extrusion. And so if we look at a meta plot of this condensin binding data, we can see that basically as we add in more and more H4 tail substitutions, so we're increasingly decompacting the local chromatin fiber structure, condensin is moving away from its normal binding sites in wild-type cells. So it seems like it's consistent with sort of extrusion right across chromatin in cells. And so the model then would be in quiescent cells in the wild-type case. You have locally compacted chromatin structure. It might not look like this drawing, but this chromatin structure really kind of represses the ability of condensin to extrude, or at least slows down condensin extrusion. But in mutants that decompact local chromatin structure, we see the presence of really more active loop extrusion. So it seems like compacted structure on the local level may really regulate loop extrusion. And so the whole model then is that between log and quiescence, condensin relocates through the genome, forms these large repressive chromatin loop domain structures. And then at the same time, H4 and probably H3 deacetylation drive the local compaction of chromatin fibers. So chromatin structure, both of these levels, is involved in repressing transcription. And additionally, we see that this change in local chromatin structure may also help to stabilize condensin localization at sort of the base of these loops. And so I think this is a really nice system for potentially validating kind of modeling, looking at chromatin structure at least either of these scales, right? Because this is such a nicely perturbable system, and because we can get very high resolution, we think, if there's something that you think might be interesting to test, we can mutate it or delete it or repress it in these cells and see what happens. Is that my time? No. Okay. Perfect. All right, so the last few minutes, I just want to touch on some of the things that we're working on now in the lab. And so we've really only been functional for about a year, so this is all pretty preliminary, but I didn't want to just talk about published data. So one of the things we're really interested in is looking at what drives condensin to kind of relocate through the genome to form these chromatin loops at gene promoters during quiescence entry. And so we have a transcription factor that overlaps with condensin binding that we think may be functioning similarly to CTCF in these cells. So we're looking a lot at that. We're also looking at the effect of transcription and potentially recruiting or affecting loop extrusion in these cells. Additionally, we're really interested in looking at the dynamics of chromatin during quiescence entry. Now, just to remind you, so this entry process actually takes a week. And so I think this is a cool opportunity where we can look basically at each day or, take very kind of lazy time points through this process to look at the dynamics of these things during this entry process. And so we have done sort of DAPI, low resolution method looking at the progression of condensation during quiescence entry. So here we're looking at G1 arrested cells and our quiescent cells. Oops, losing my pointer here. We can see that we get, we have very condensed chromatin in these quiescent cells. And we also looked at day 5 and day 6 going into quiescence entry. So we can see that this condensation is really a gradual process that seems to be taking basically the whole week, right? Which is very odd because the week is very long in yeast time, right? We've also done condensin ChIP-seq each day during entry so far in these purified quiescent cells. So here I'm just showing kind of a genome browser view of part of chromosome 2, looking at this condensin ChIP-seq data. And so if we look progressively from day 3 all the way to day 7, which is what we're calling quiescence, we really can see sort of the gradual accumulation of condensin at its normal kind of binding sites in quiescence. So we think this will really give us a chance to kind of temporally dissect the different factors that are involved in bringing condensin here, and maybe also look at the relationship between extrusion and transcriptional repression. All right, so the last thing that we're currently working on, I'm still working on expanding the lab, is really kind of looking at this question of how do chromatin structures translate into genomics data or into micro-C data. And so I think really a lot of sort of studies of chromatin structure based on high-C or micro-C data tend to focus in on these loop structures, right? And I think part of that is that these loops are very interesting and important. The other part of it is really that a loop is one of the most obvious things to kind of interpret from this kind of data. I mean, it's easy to see a dot that's far away from the local interactions and say, oh yeah, that's a loop. Whereas all the other things that are located in these maps really are much more kind of difficult to understand. And so, for example, if we were to have something like a 30 nanometer fiber in cells, it's not entirely clear how this kind of structure would translate into context, right? Because, although we might expect that we would have this sort of zigzag contact pattern where you have a nucleosome interacting with one, every other nucleosome, so N plus 2. We have a state where we have cross-linking, so it's not clear how this cross-linking would sort of affect that pattern of interactions. Additionally, we're looking at a whole population of cells, right? So again, not clear exactly what's going on at these kind of other levels. And so one way we're working on approaching this is to kind of develop this whole genome-wide reconstitution system from Philip Korber's lab. And so for those of you who are not familiar with this, in this system, they have a library of plasmids that basically incorporate the entire yeast genome. You can reconstitute chromatin on this library through salt dialysis, and then you can add in, nuclear extract or any proteins of interest, and then do genomics on this system. And so the Korber lab has really worked out very nicely, kind of how to make this system look as close to in vivo chromatin as possible. And so essentially, we're looking at native MNA-seq data on cellular chromatin. And so we see kind of the typical pattern of nucleosome positioning that we expect. So if we just do salt gradient dialysis on this system, right, we can kind of recapitulate our nucleosome depleted regions, but positioning doesn't look very good. But as we add in other factors, and particularly NO80, the chromatin remodeling enzyme, and additionally these sort of general regulatory transcription factors, we can start to get really nice nucleosome positioning here. And so we're starting to use this system with the goal of doing micro-C on the system, so getting a sense of how these kind of contexts translate, and then comparing this to our data from cells to see if we can start to reconstitute this quiescent cell chromatin kind of from the bottom up. Okay, I'm good. All right, so I just want to thank several people, and so a lot of this preliminary work was done by members of my lab, some of which were rotation students. Some of this work was done by Bill Dakota and Jeff McKnight in Toshi Tsukiyama's lab. Sung Su and Jay Shinderi's lab, and Dejan Lin and Bill Noble's lab did some of the analysis. And then Stephanie and Tamara, of course, did the modeling. And Po Yin Yin did some STEM tomography that I didn't have time to talk about today. That's it. Thank you. Right. Yeah, I don't remember seeing anything special about the mat locus and the quiescent cells. I think basically, it's all kind of compacted generally. How SIR proteins are involved in changing in this quiescent system, I haven't specifically looked. There is some cell biology data from Isabel Segov's lab kind of looking at SIR localization in these cells, but I haven't really seen anything. Yeah, so they're doing sort of confocal microscopy. So they have evidence that actually telomeres move from the nuclear periphery and come to the interior of the nucleus in these cells, and that's dependent on SIR proteins. I think that's about as much as we know right now. Mm-hmm. Yeah, it does get tighter. So we can see in MNA-seq data that the positions get a little bit tighter, and the nucleosome depleted regions also get tighter. We can look at H3 ChIP-seq data and see occupancy also goes up a little bit. So I don't think it's a huge difference unless we're talking about the nucleosome depleted region, but it's definitely on the order of maybe 5 base pairs. Something. It's very heteromorphic, right? Mm-hmm. Yes. Yeah. So you don't see changes in nucleosome density in the mutants. I haven't looked at positioning, so I don't know the small amount of decrease that we see in the linker length. I don't know if that's preserved in the mutants or not. We should do that though. Oh. Yeah, I mean, maybe if we do TSA or something where we can actually kind of time it out, right? I think it, right, right. I mean, if someone wanted to look at that, maybe in a collaboration, that would be great. Mhm. Yeah. Um, mhm. Well, that's something we think about a lot. I mean, I think the Daffy staining, I mean, we get sort of a measurement in terms of volume, right? I do have a student who's been really working in order to, because it'd be nice if we could quantify compaction on a gene level, right? If we really want to look at transcription. And so we're looking at contacts, basically, across genes. And so contacts per base pair or contacts per nucleosome would really be the measurement. Right. So I mean, that's a linear thing, right? If you have a better idea, though, I'm happy to hear it. That's not fair. Thank you.